Welcome to the Deep Dive. We're your shortcut, really, to getting properly informed without, getting totally bogged down in reading everything yourself. We distill it all down, find those surprising facts, add just enough, maybe humor to keep you listening. We guide you through the dense stuff.
Yeah, cut through the noise.
Exactly. And today, we're tackling the world of medical science. Specifically, we're doing a deep dive into the evidence around a pretty novel compound, Rilzabrutinib.
Right.
Our focus is going to be its role in immune thrombocytopenia, or ITP. And we've tailored this whole exploration specifically for you, our listener, especially if you're maybe aiming to build a...
We'll cover the strengths, the limitations of what we know now, and really critically for your purposes, the best directions for future research. It's all about giving you that comprehensive picture, but, in a way that's easy to grasp and use.
Makes sense. A solid foundation.
Okay, so before we dive headfirst into Rilzabrutinib itself, maybe you could lay the groundwork for us. What exactly is ITP?
Sure. It's important context. So, immune thrombocytopenia, ITP, is a rare condition, but it has a really profound impact. It's an autoimmune bleeding disorder.
Autoimmune meaning the body attacks itself.
Exactly. The immune system basically gets confused. It mistakenly sees... tiny little spots on the skin, petechiae, up to more severe internal bleeding. And there's also this really debilitating fatigue. It often gets overlooked, but it massively impacts quality of life.
Fatigue, really? That's interesting.
Oh yes, it's a huge factor. Now look, many patients might have relatively mild symptoms, but we have to remember serious, even fatal bleeding can happen.
Especially in certain groups.
Yeah, the risk is definitely higher in older patients, or in people whose counts drop really low. We usually use the threshold of below 30 times 10 to the 9th per millimeter cubed. So understanding this, the mechanism and the real impact, it's fundamental, especially if you're framing that problem statement for a grant. You need to show the need. Yes, the thrombopoietin receptor agonists, and there's fostamatinib too, plus rituximab is used quite a bit, and of course corticosteroids are often the first thing tried.
Okay, so quite a few options.
Yeah,
It sounds like there's a but coming.
there is, yeah. Despite all these options, there's still this significant minority of patients who just don't respond adequately. Their disease is resistant.
And managing them must be incredibly tough.
It's really challenging. It feels like an uphill battle for them and their doctors. They might end up trying experimental combinations, or they're stuck on chronic corticosteroids long term.
Which have their own serious side effects.
Absolutely. Osteoporosis, diabetes, infections. It's not ideal. That's a key point. Raising the count is one thing, but we often see high relapse rates when treatment stops or tapers. Patients cycle through therapies. There are side effects, obviously, sometimes making long-term tolerability a real problem. People stop treatment, and maybe most importantly, these treatments often don't fully address the health-related quality of life issues, especially that fatigue we mentioned. It can be just as bad as the bleeding for daily life.
So it's not just about the lab numbers.
Not at all. It's about the patient's actual experience, their ability to function day to day.
Right. So it really hammers home that there's this high unmet therapeutic need, particularly for those with
Now, on Rilzabrutinib itself, we know it's novel, but what is it fundamental? How does it work at that molecular level?
Right, the nuts and bolts. So Rilzabrutinib is an oral drug, which is a plus, and it's a reversible covalent Bruton tyrosine kinase inhibitor, BTK inhibitor for short.
BTK, what role does that play?
Think of BTK as a really key signaling molecule inside immune cells. It's like a switch downstream of several important receptors, B cell receptors,
FC gamma receptors, FC epsilon receptors.
Okay.
So BTK is really influential in things like B cell development, antibody production, which is crucial in ITP because you have these
preclinical studies really map this out.
So, what does it actually do?
Well, it hits several key parts of the ITP disease process. It inhibits B cell activation, so you damp down that initial immune response. That, in turn, reduces the production of those harmful autoantibodies that target platelets.
Okay, stops the antibody production.
And importantly, it interrupts the platelet destruction that phagocytosis mediated by the Fc gamma receptors, mainly in the spleen and liver where most platelets get cleared in ITP.
So it protects the platelets that are there.
Exactly. And it also seems
to inhibit inflammatory pathways that contribute to the general immune chaos in ITP. So it's attacking the problem from multiple angles, not just one symptom, but the underlying.
irreversible.
Which sounds strong.
It is strong, which is good for potency, but if the drug accidentally binds to the wrong target, an off-target effect, that bond is permanent too, which can lead to persistent side effects, or the effect lasts even after the drug is cleared.
I see.
Rilzabrutinib, though, forms a reversible covalent bond. Think of it like a key fitting perfectly, but held by a really strong yet temporary magnet instead of being glued in place.
Okay.
This allows for really long target engagement. It maintains over 80%
BTK occupancy within the first hour and keeps that high level for over 24 hours. That sustained engagement is crucial for the therapeutic effect, keeping those disease pathways.
Potentially, yes. There's also a theoretical advantage in reducing the risk of rare idiosyncratic drug toxicity compared to irreversible ones. But even though it's reversible, it still provides that sustained effect because it unbinds from the target very slowly at a slow off rate. It's a sophisticated way to try and get the best of both worlds. High potency and a potentially better safety profile.
That is clever.
Yeah.
Okay, so we understand the design, the mechanism.
Right.
But the million-dollar question is
always, does it work in people? How did this theory play out in clinical trials? Let's dig into the evidence.
Right, the clinical data. But first, it's really important to remember the challenge of ITP.
Okay, understood. So let's start with the big one. You mentioned the LNA3 phase 3 trial. Who it all? 2025. Pivotal, randomized, double-blind, placebo-controlled. Tell us about that. Who was in this trial?
Yeah, LNA3 was a major effort, global scale. It enrolled 202 adult patients across 26 countries. 133 got Rilzabrutinib. 69 got placebo.
And the patient type?
Adults 18 or older with persistent or chronic primary ITP. But here's the critical part, and you need this
for your grant rationale. These were heavily pretreated patients.
See, and they tried lots of things before.
Lots of things. The median time they'd had ITP was 8.1 years in the Rilzabrutinib group.
Absolutely. To even get into the trial, they needed platelet counts below 30 at screening, and they had to have shown they didn't get a sustained response from other therapies or couldn't tolerate them. This was a group where existing options just weren't cutting it.
And the study design, double-blind for how long?
Double-blind treatment went for up to 24 weeks, so neither patients nor doctors knew who got what. A standard way to minimize bias. Everything was assessed for efficacy and safety.
And many people drop out.
There was a notable rate of discontinuation,
Particularly for lack of platelet response, but it was much higher in the placebo group. 80% stopped for that reason compared to 41% in the Rilzabrutinib group. That
efficacy findings were definitely encouraging, especially the primary endpoint. It met that endpoint, which was durable response. 23% of the Rilzabrutinib patients achieved this. That's 31 people.
Compared to placebo.
Compared to 0% in the placebo group, none of the 69 placebo patients hit that mark, and statistically the difference was highly significant. The p-value is less than 0.0001.
Which means it's very unlikely to be chance.
Extremely unlikely. No durable response had a strict
definition. Getting a platelet count of 50 or more on at least 4 out of 6 visits between weeks 14 and 24 without needing rescue meds or stopping treatment.
And
on placebo. That fast response is really important when dealing with bleeding risk.
And the actual platelet levels, did they get much higher?
In those who had a durable response on Rilzabrutinib, the median platelet counts really climbed nicely. They got above 30 by week 2, above 50 by week 3, and then over 100 by week 13, staying up there afterwards.
And counts over 50 or 100 are clinically meaningful.
Hugely. Getting over 50 significantly cuts the risk of serious bleeding. Getting over 100 generally means bleeding risk is minimal. Even for procedures, it's a big deal for patients.
compared to placebo.
In contrast, the median counts in the placebo group generally stayed below 30 throughout the study. Really highlights the drug's effect.
Any other benefits, like needing fewer rescue treatments?
Absolutely, solid data. Now, we talked earlier about how debilitating fatigue and poor quality of life can be in ITP. Did ILLIN-A3 look beyond the platelet counts at how patients actually felt? Were patient-reported outcomes included?
They were, yes, and that's increasingly important. As you say, we can't just focus on the lab values. There was an abstract specifically from ILLIN-A3 reporting improved health-related quality of life, HRQOL, with oral Bruton tyrosine kinase inhibitor, BTK,
Rilzabrutinib versus placebo.
That sounds really positive.
It is, because as we discussed, ITP isn't just about bruising. It's the fatigue, the impact on daily function, emotional well-being.
benefit is key.
Incredibly important. Okay, efficacy looked promising, quality of life improving.
Yeah.
But safety is always the other side of the coin. How did Rilzabrutinib fare in ILLIN-A3? Any major red flags or side effects?
Overall, the safety picture looked pretty good. Rilzabrutinib was generally well tolerated.
What about common side effects, the treatment-emergent adverse events, or TEAs?
Right, the TEAs that occurred in at least 5% of patients in either group. Here's where it gets a bit, well, curious actually. Some common things were reported more often in the placebo group.
More often in placebo. Like what?
Increased tendency to bruise, 11% on Rilzabrutinib, but 25% on placebo. Myopathy, muscle issues, 15% sphinxis, 23%. Insomnia, 9%.
Good question. It raises some points for, epidemiological thinking? Is it just background noise, the symptoms of chronic ITP itself bubbling up? Is it maybe a nocebo effect in the placebo group, people feeling worse because they expect side effects? Or just the general symptom burden in this very sick population? It definitely warrants looking into more in real-world data later.
What about serious adverse events, SAEs?
SAEs did occur in both groups, which is unusual in trials like this. In the
Rilzabrutinib arm, things like pneumonia, neutropenia, peripheral embolism, COVID-19 pneumonia, bacterial meningitis, septic shock were reported.
Any deaths?
There was one death
About 6% of Rilzabrutinib patients discontinued because of adverse events, compared to none in the placebo group for that reason. But remember, far more placebo patients, 80% stopped because the drug wasn't working for their platelets, compared to 41% in the Rilzabrutinib group. So while the case did cause some dropouts, the overall tolerability seems quite reasonable, especially given the higher rates of some AEs in the placebo arm and the efficacy difference.
Okay, that gives us a really detailed picture of Elune-3.
But drug development is a journey. What about before this big phase 3 trial? Did earlier studies give hints that Rilzabrutinib might work?
Oh, absolutely. You always start smaller. There was an
twice daily achieved the primary endpoint.
Which was platelet response.
Right, defined as getting platelets over 50 and having at least a 20-point increase from baseline. And one really striking thing from that early study was how fast it seemed to work. The median time to get platelets up to 50 was just 11.5 days. That's quick and really valuable if someone's actively bleeding or at high risk.
What about safety in that early trial? Any major concerns like bleeding or clotting?
That was a key safety finding. They specifically noted no cases with significant bleeding and thrombosis were observed.
That's huge for an ITP drug. You don't want the treatment causing bleeding, and thrombosis or clotting can sometimes be a concern with immune modulation.
Were there any side effects?
There were some great...
Okay, that makes sense. We've talked about how it works in ITP patients. But what about how the drug itself behaves in the body? What do we know from pharmacokinetic studies, maybe in healthy volunteers? How is it absorbed, eliminated? Does it interact with other drugs? That's all crucial for actual clinical use.
Absolutely critical. Understanding the PK, the pharmacokinetics, is fundamental. And yes, there were studies in healthy volunteers looking specifically at this. Papers by Ukpanar and colleagues, Rask Madsen, they detailed the
PK profile.
What did they find?
It seems to be absorbed rapidly after you take the pill. The peak concentration in the blood, the Tmax, usually happens around 1 to 3 hours later. Elimination looked...
That's an important point. They noted high between subject variability, meaning different people can have quite different levels of the drug in their system, even on the same dose, and also high within subject variability. So even the same person might have different levels on different days.
What does that mean, practically?
It means dosing might be a bit more complex in the real world. It might not be a one-size-fits-all drug, maybe needing more careful monitoring or adjustment for some individuals compared to a drug with
very predictable PK.
What about interactions with other medications, DDIs?
Yes, very important. They found a really significant interaction with ritonavir. Ritonavir is a...
to prevent potential overdose and side effects.
Did it interact with everything?
No. For example, a co-administration with quinidine, a different type of drug, showed no significant change in Rilzabrutinib's PK. So you need to know which
particularly cardiac safety.
That's a really positive point here. They did a thorough QTC study looking specifically at potential effects on heart rhythm. They found no QTC prolongation with Rilzabrutinib, even at doses higher than the therapeutic ones.
Why is that significant?
Because some other
BTK inhibitors and other drugs in general can affect the QTC interval, which can increase the risk of serious arrhythmias. So the lack of this effect with Rilzabrutinib is a
That PK profile is really useful context. Now, let's zoom out a bit. We focused on the clinical trials. What do these studies tell us, if anything, about the broader epidemiology of ITP itself, or how Rilzabrutinib might be used in the wider population? Are we getting insights into prevalence, incidence, real-world trends?
That's where we need to be careful. The literature we're discussing today is almost entirely focused on these controlled clinical trials. They're designed to test the drug under specific conditions in selected patients.
So not broad population studies.
No, they don't really give us new data on the overall prevalence or incidence of ITP in the general population. We know ITP is rare, maybe around 9.5.
Level data from trials alone.
What about real-world use of Rilzabrutinib? Do these sources tell us anything about how it's being used outside of trials, maybe post-approval in some regions?
No, the sources we have don't provide that information. They focus on the trial data leading up to potential approval. So there's currently a knowledge gap regarding its real-world use, its uptake in actual clinical practice, or its long-term performance in diverse unselected patient populations.
So no population-level trends yet from this data?
Not from these sources, no.
That kind of real-world epidemiological insight would typically come later from post-marketing studies, registries, or database analyses once the drug is more established. It's a clear
broad population data isn't there, the trials definitely did focus on a key subgroup. The one consistently targeted, especially in LUMINA-3 in the phase 12, was that heavily pretreated, persistent or chronic, often refractory ITP patient population.
The really difficult to treat ones we talked about.
Exactly. These are the patients who've run out of good options. The LNA3 characteristics highlighted this. Long ITP duration, multiple prior therapies including steroids, TPO arrays, even splenectomy.
That is a specific high-need subgroup they deliberately studied.
And did the drug work consistently within that subgroup?
Well, the LNA3 paper mentioned the durable response was
experts point out the limited ability to stratify subjects of defined subgroups because the overall numbers are just too small to slice the data too many ways and still have statistical power. It's hard to drill down into really granular subgroups based on specific risk factors. And there's that other challenge we touched on, really nailing down who is truly refractory versus having maybe something else going on that looks like ITP but won't respond to ITP drugs. That inherent heterogeneity makes subgroup analysis tricky in trials and is
definitely an area for future epidemiological work, maybe using real-world data or registries.
Okay, so bringing this all together now, for a listener, maybe sketching out that grant proposal.
All right, the summary for the proposal. The strengths are pretty compelling. First, and this is key for justifying research, it addresses a critical unmet need. It's being developed for refractory ITP patients who are struggling with current options, facing severe disease, poor quality of life, higher mortality. Targeting this group has high impact potential.
Makes sense.
Second, the novel mechanism of action, that reversible covalent BTK inhibition, the multi-immune modulation. It's a targeted approach hitting the disease pathophysiology
from multiple angles. The design aiming for high selectivity and potentially reduced off-target toxicity is a real plus compared to broader immunosuppressant.
Seen earlier is also a significant advantage. You have positive data to build on.
And safety.
Generally, a favorable safety profile. Well tolerated overall in the trials. Crucially, no major signals for drug-induced severe bleeding or thrombosis, which is vital in ITP. And that clean QTC study adds reassurance regarding cardiac safety. That combination of efficacy and safety is powerful.
Okay, those are strong selling points. But a balanced proposal also needs to acknowledge the weaknesses or gaps. What are the main limitations or methodological concerns someone should address?
Absolutely. Showing you
Understand the limitations is critical for credibility. The biggest, most unavoidable one is the rarity of ITP itself. This just fundamentally limits trial sizes, statistical power, the ability.
Defining who is refractory.
Yes, the lack of a consistent definition for refractory ITP is a field-wide challenge. It makes comparing results across different studies difficult because the patient populations might not be quite the same. And distinguishing truly refractory ITP from other conditions remains tough. This affects patient selection and interpretation.
Any other limitations?
The pharmacokinetic variability we discussed high between subject and within subject variability could make dosing more complex in the real world. The drug interaction with ritonavir is a practical limitation requiring careful management.
Right.
And finally, the big one for the future. Limited real-world data. Almost everything we know is from controlled trials. We don't yet...
This is where you propose solutions and next steps. Several areas stand out. First, we need to enhance study design strategies specifically for rare diseases like ITP. Consider things like umbrella trials. These are master protocols where you can test multiple new drugs or arms under one common control group, making it faster and more efficient to get answers compared to setting up separate trials for everything.
That sounds smart.
It is. Also, designing trials to smartly integrate different patient groups, maybe refractory patients,
alongside those with contraindications to standard care, could help assess efficacy more broadly without needing huge separate studies for each niche. And pursuing more observational...
Big improvements needed there. We really need to improve data collection and reporting, especially focusing on what matters to patients. Advocating for prospective registries, ideally linked with biobanking collecting samples for future research is crucial. Models like the French Ceravance registry show how this can work.
What's the benefit of registries?
They capture real-world treatment use, including off-label, allow long-term follow-up of large groups, and the biobanking enables studies into disease mechanisms and heterogeneity. It's invaluable for rare diseases.
And alongside this, we must consistently include patient-reported outcomes, PROs, and quality of life, QOL, measures as core endpoints in trials, not just secondary.
Fourth, dive deeper into investigating biological heterogeneity. Why are some patients refractory? It's likely biological differences. Future research needs to integrate more biological assessments, genetics, proteomics to identify subgroups who might respond differently. This is the path towards more personalized medicine in ITP.
Find the right drug for the right patient subgroup.
Precisely. Fifth, the field needs to continue working on refining the definition of refractory ITP.
Getting clearer, standardized definitions will make research much more consistent and comparable across studies.
And finally,
Always balance the risk. As we get more potent therapy,
Okay, so let's try and bring this all together. This deep dive really highlights Rilzabrutinib as, well, a genuinely promising new player. It's an oral, reversible BTK inhibitor showing strong efficacy and a decent safety profile, especially in those heavily pretreated ITP patients who have limited options. It seems to directly target a critical unmet need.
That sums it up well. Significant progress has definitely been made, but the journey in rare diseases like ITP is absolutely ongoing. The focus now is sharpening more precise
targeting, really listening to patient-reported outcomes, and gathering that vital, real-world evidence to see how these drugs perform long-term in everyday practice.
It's a constantly evolving picture, so a final thought for you, our listener, to mull over. As we see these highly targeted therapies like Rilzabrutinib emerge, what new questions does this raise for you about the future of personalized medicine, especially in these rare, complex conditions? And perhaps more fundamentally, how do we ensure that our relentless drive for efficacy, for better platelet counts, never overshadows the importance of holistic well-being and the long-term quality of life for every single patient? It's about more than just the numbers.
thank you for joining us for this deep dive.